In this segment on cell membrane transport, now we are going to be focusing on osmosis. So remember, osmosis is another form of passive transport. There's still 2 more videos to go on membrane transport, primary active transport and secondary active transport. But let's go ahead and focus on osmosis. So when you think about osmosis, there are some terms that are important for you to understand, and that is, what is a solution? So a solution is a solute plus a solvent. So if you mix the 2 together, you form a solution. But now you need to understand what a solute is versus a solvent. So the solvent is the dissolving medium. So in human physiology, our dissolving medium will always be water because we're dealing with the biological system. The solute, however, can vary. The solute is what is dissolved in the solvent or disassociates in the solvent. So if you look at the 2 beakers in the right-hand side of your screen, it says which side is considered a solution, A or B. So the legend is down below and it tells you what color the water molecules are versus the solutes. So side B is considered your solution because it consists of both solutes and solvent. Okay, so when we talk about osmosis then, osmosis is strictly the diffusion of water. So anytime we're talking about osmosis, you need to think about water diffusion. Water is going to be moving from a high concentration of water toward a low concentration of water, which is the same thing as saying that water will move from a low concentration of solute toward a high concentration of solute. And osmosis requires an aquaporin. So an aquaporin is a specialized sort of channel. And so looking at the figure to the right, and the question asks you which side has the highest concentration of water. And so if we look at side A has a higher concentration of water and a lower concentration of solute. Side B has a lower concentration of water and a higher concentration of solute. Okay, so side A has a higher concentration of water. Okay, so if you pause the video, then you can answer this question. And the question is, which direction is water going to move, assuming that you have a non-permeating solute? A non-permeating solute means that the solute is not permeable to the membrane. And so here you have a beaker and it's separated by a membrane. But the solutes are not permeable. They cannot pass through that membrane. So we want to know, well, which direction is water going to move then? So you could pause the video to answer this question. So always remember, when water moves, it moves from a high concentration of water toward a lower concentration of water. So water then is moving from side 1 to side 2. The result of that is shown here. And you can see how the water level in side 1 went down and the water level in side 2 went up. So the volumes changed. That's an important concept. Volumes change when water diffuses. Okay, so now let's start tying this into the term tonicity. So tonicity describes the behavior of cells when you place them in a solution. And so when we say the behavior of the cells, we're talking about whether or not that cell may shrink or will that cell expand and then burst. So different types of solutions can do this and can change the behaviors of these cells. So the solutions that we want to discuss are hypotonic, hypertonic, and isotonic. So let's go through each of these solutions and think about how that will change the cell size. So let's begin with a hypotonic solution. So here you have a cell that's been placed inside of a beaker. When you work these problems, it's going to be very important for you to think through the problems and rather than memorize, okay? Because there's ways that these problems can be written such that memorization won't do you any good. So this is how you work through the problems and how you think through them, and this way you never have to memorize anything. So we know hypo means less. Tonic means solute, okay? And here we're talking about a hypotonic solution. So the solution is in the beaker. So it's hypotonic. So that means I have a low concentration of solute. which then tells me in the cell, because I have to compare the solute concentration to something else, and here I'm comparing it to the cell. So in the cell, I have a high concentration of solute. Now we want to compare that to the water concentration. If there's less solute present, then that means there's more water. And then just the opposite condition within the cell, a lower concentration of water in the cell. So now you've set up your problem, and now what you can do is determine the direction of water movement. So we would assume that there's an aquaporin present here, and if there's an aquaporin present, then water is going to move from a high concentration of water toward a low concentration of water, meaning water will move into your cell. So again, don't memorize hypotonic solution means water will move into the cell, because then you're just memorizing it and you're not able to think through it and walk your way through it. And so you'll wind up missing questions because of the twists and turns that can be done to these types of problems. Then there's a hypertonic solution. So what I would suggest is that you pause the video, set up this problem, and tell me then which direction water is going to move. Now before I forget, let's go back to the previous slide for a minute. If water is going to be diffusing into the cell, how does that affect the volume of that cell? So this cell can get bigger and bigger and bigger, right? It can expand and expand and expand until eventually this cell will burst. So this cell will lyse. So now let's go back to the hypertonic solution. Okay, so if you've already paused the video and set up the problem, then you can continue on with the video. So the hypertonic solution then tells me that I have a high concentration of solute, right? Because hyper means high or lots of. Tonic again means solute, and we're talking about the solution. So that's where I'm getting a high concentration of solutes in the beaker. All right, and then in the cell, I have a low concentration of solute with respect to the solution. And now we determine where we have a higher concentration of water. Well, if we have less solute, that means that we have more water. And if we have high solute, that means we have less water. And I'm going to put solvent here just so that you remember solvent and water are the same thing. So now which direction is water going to diffuse, assuming an aquaporin is present? So water then will diffuse out of the cell, moving from a high concentration of water toward a low concentration of water. So since the cell is actually losing water, that means that this cell is going to shrink or shrivel. Then we have isotonic solutions. So iso means same. Tonic again means solute. So what that means then is that we have the same amount of solute, the same concentration of solute in the solution as we do in our cell. So if we have the same concentration of solute in both locations, then we must have the same concentration of solvent, water, in the solution as well as the cell. And if we assume an aquaporin is present, do we have a net movement of water? Okay, the answer is no, right? Water cannot move. There's no net movement of water because there's no gradient for the water to move. So in an isotonic solution, water is not diffusing. There's no net movement of water. Okay, so then a little question for you. Would it be appropriate to describe a cell as hypotonic, hypertonic, or isotonic? Or can we only use those terms to describe solutions? And the answer to that is absolutely. You can say a cell is hypertonic to a solution. You can say a cell is hypotonic to a solution. Notice I'm always comparing it to something. It's hypertonic. The cell is hypertonic to a solution. Okay? And so this is why I don't want you to memorize what happens in hypertonic solutions or hypotonic solutions, because then what happens if I turn it around and I say, oh, the cell is hypotonic or the cell is hypertonic to the solution? Now that's going to blow your mind, and now you're going to have to try to keep things straight. So it's not difficult to set up these problems and work through them. So that's what I highly suggest for you. Okay, so here's a practice problem for you. And so what I'd like for you to do is to pause the video and work this problem. So here we have a cell, and it's placed in a saltwater solution of 0.2 millimolar sodium chloride. Now, in these problems, it's always going to be important for you to draw, okay? So I'm just going to take a cell and I'm going to put it into my solution. So here's my cell, okay? And I'm putting it into a saltwater solution that has 0.2 millimoles of sodium chloride. Okay. Neither sodium nor chloride can permeate the plasma membrane, meaning those ions are not going to be diffusing. So we want to know, well, what's going to happen to the cell? And I gave you a little hint here. The osmolarity of sodium chloride in the intracellular fluid is less than 0.2 millimoles. Okay, so now we're dealing with osmosis, correct? And now what we need to do is convert to osmoles, or milliosmoles is fine as well. So sodium chloride will disassociate in water. And when it disassociates in water, you get 0.2 sodium. You get 0.2 of chloride ions. So that means we're going to have 0.4 milliosmoles of sodium chloride. Now, inside the cell, that means we're going to have less than 0.4 milliosmoles, right, of sodium chloride. So now my question to you is, where is there more water? Okay. So in other words, if you have less solute, you're going to have more water, right? So we have less solute in the cell, which means we have more water in the cell, less water in the solution. So that means our water is going to diffuse out of that cell. In this particular problem, we already know it's an osmosis question. However, if you were working this on an exam or in the laboratory, you would be told that an aquaporin is present. So we could actually add to this. Assume an aquaporin is present. Okay, and that'll let you know then that water is capable of diffusing if there's an aquaporin present. Okay, so the answer then, if water's moving out of the cell's going to shrink because the salt water, the solution, is hypertonic. There's more solute in the solution than the cell.